Welcome to the Cell Communication series. This video focuses on cell signaling. At the end of the lecture, students should be able to understand how cells communicate with one another. They should differentiate between the types of cell signaling and explain the 3 stages of cell communication, in addition to indicating how receptors are activated. All cellular communication exhibits certain features. First, there must be a detection of the environmental signal. This occurs via a cell receptor. Second, the cell must be able to interpret that signal, which occurs through conformational change in the receptor site. Third, the cell responds to the signal within its internal environment. Finally, when necessary, the cell will put out or produce its own signal to communicate to other cells. Signals are often used to influence properties of cells, both internally and externally. They're used to coordinate the activities of multicellular organisms, to change the conformation of a receptor, and can even lead to apoptosis or cell death. One way cells use signaling is to respond to changes in their external environment. These changes in the environment can easily alter their ability to survive, which happens regularly with changes in temperature, pH, salinity, and pressure. In this example, glucose becomes abundant in the environment external to a yeast cell. It is detected by the yeast's glucose receptor. The glucose binds to the receptor, calling for a cellular response. In this case, since glucose is food for the yeast cell, the yeast will increase the number of glucose transporters throughout its membrane. This takes the additional glucose through into the cell while generating additional metabolic enzymes to process the glucose most efficiently to make energy. A side note, the circle around the yeast's membrane is an organelle that, in conjunction with the membrane, forms the cell envelope. The envelope is sometimes referred to as a cell wall because it does provide structure, strength, and protection for the yeast. The wall, as with fungi, does contain chitin, and it's porous, which allows certain materials to cross. Things like glucose can easily pass through and interact with the membrane. Another way signals are used is for cell-to-cell communication. There are multiple forms that cells use for communication that differ widely based on the distance between the cells. Each form of intercellular or outside of the cell communication requires a cell to produce a signal and another targeted cell to receive it. The first form of communication is direct intercellular communication. In multicellular organisms, the cells that are all close together have small channels between them, allowing them to share materials between their cytoplasm. These adjacent cells also pass signals through the channels. In organ systems, these cells communicate important cellular functions to work together in harmony. The next style of communication is contact-dependent signaling. This type of communication requires the signaling cell and the receiving cell to come into contact with one another. Neurons use this type of communication to create their synapses between them so that they are close enough to transfer messages along their pathways. Sometimes cells need to send themselves, along with other cells of the same type, signals. These are autocrine signals, auto meaning self. One way to control cell or tissue growth is to use autocrine signals, which tell other cells there are too many of their type of cell out there. They should stop undergoing or turn off cell division. Paracrine signaling, by contrast, is an alert to other cells. It doesn't affect the transmitting cell. Paracrine signaling is broken down very quickly and therefore can only be used in communication with target cells that are in close contact. The nervous system uses paracrine signaling to pass information in the form of neurotransmitters from one cell through the synaptic cleft to the target cell. The fifth form of communication is through endocrine signaling. The endocrine system uses signals, called hormones, to send messages between various parts of the body. In this way, it uses long-distance communication. Therefore, signals have a long life. Endocrine hormones use the bloodstream as a travel mechanism between all the parts of the body. Once a signal is transmitted, a target cell needs to receive the signal. In order to do this, it follows a three-step process known as the signal transduction pathway. These stages are activation, transduction, and response. First, the signal molecule binds to the receptor site on the target cell. This causes the receptor to change its conformation and the protein function for the cell. Next, the signal is transduced or converted into a different signal targeted inside the cell. A series of proteins undergoing these changes from the signal transduction pathway can produce intracellular or within the cell signals. Finally, the cell responds by altering the targeted proteins. The target proteins generally respond in three ways. One mechanism is by adjusting enzymatic activity. When we need energy, hormones signal to break down carbs and make ATP. A second response cells have affects the structural proteins in the cell. As amoeba move their pseudopods or false feet, by rearranging their cytoskeleton, they change their shape and thereby change their direction. A third response mechanism, via transcription factors, allows us to make proteins that express genes or certain characteristic traits. These factors can influence gene expression by turning it on or off at various points or in various environments. Receptors are activated when they receive a chemical signal from the environment. Many signaling molecules are steroids or proteins that bind to receptors, like the binding of an enzyme with its substrate. These lock and key mechanisms allow for only specially shaped signals to bind with each receptor. The signal is called a ligand, and it moves in the environment until such time that the ligand rotates into a specific conformation for the receptor acceptance to occur. As the ligand's specific orientation is reached, while it collides with the receptor, a ligand-receptor complex is formed. Although it takes lots of energy and specificity even for the appropriate ligand, this association occurs relatively quickly. The ligands do not bind covalently to the receptor. When ligands are present in the environment and bind to their receptors, a conformational change occurs to the receptor. The changes in the receptor activate the receptor and allow it to initiate a cellular response. When the ligand disengages from the receptor, the ligand-receptor complex will dissociate. This causes the receptor to revert back to its original conformation, in which it is no longer activated. Students should be able to understand the four features of cell-to-cell communication. They should be able to differentiate between types of cell signaling. They should be able to differentiate between types of signal relaying. They should be able to explain three stages of cell communication and discuss how receptors are activated.